Good afternoon. This is Nelly Poyak from Dana Farber. Welcome to our DFHCC Connect Science seminar series. Thank you for joining. A reminder that the slide presentations will be uploaded to the website that's shown on the first slide, and we have a past presentations link, so please go there, and then we have a list of the past presentations that, when the speaker agreed to recording, and then you can view the slides in case you missed it today. Also, as a reminder, please put your questions in a Q&A, and we answer as many questions as we can at the end of the talk, and the speaker will have access to the questions later on and can get back to you. So with that, I'd like to welcome today's speaker, Sandy McAllister. She's one of our colleagues from Brigham and Women's Hospital, and Sandy did her postdoctoral training with Bob Weinberg and published a very exciting and beautiful study showing how primary tumors can have systemic effects in influencing immune environment and other, environment of the distant sites. And she continued this in kind of work in her own lab, and also expanding it to looking at the impact of age on the microenvironment and how that influences breast cancer progression. So today, the title of her talk is Time and Age, the impact of age on the breast cancer tumor immune microenvironment and therapeutic responses. So thank you, Sandy, for joining us today, and we're looking forward to your talk.
Well, thank you, Nelly, for that lovely introduction and the invitation to speak with you all today. I'm hoping you can hear me. Can someone give me a thumbs up?
Okay. Yep, sounds great.
Hear you. Yeah.
Excellent. Well, it's hard to find words right now, but I guess I would like to thank all of you for tuning in on a time that's otherwise very distracting. And no matter how you cast your ballot, I hope you're finding peace and some solace during this crazy time that hopefully we'll be moving forward together with more unity. And I really appreciate this community that we're in. So thank you very much for that collaborative and collegiality. With that, I'd like to discuss what our lab has been thinking about for quite a period of time now. And it revolves around our studies of breast cancer. Unfortunately, we know that breast cancer accounts for about 40,000 deaths per year in the United States alone, and that breast cancer affects women of all ages. In particular, we think that increased age is associated with an increased risk of breast cancer, but that older patients experience comorbidities and treatment-associated complications that makes the disease even worse for them. Likewise, in younger patients, a young age at diagnosis, for instance, under the age of 40, is associated with higher recurrence rates, and younger women are more likely to develop the more aggressive types of breast cancer, such as triple-negative breast cancer. And for both ends of the age spectrum, there are unique emotional considerations and treatment considerations such that there are age-dependent or age-specific types of approaches that we need to take when treating and caring for these patients. And here, if we look across the age spectrum at the numbers of cases, whether it's ductal carcinoma in situ or invasive, we can see the numbers of cases across the age spectrum and the number of deaths. If we look particularly at this group that's over the age of 60, what we see is that that age group accounts for the vast majority of cases and deaths associated with breast cancer. If we look, though, at the proportion of the patients that are accrued into clinical trials by age, what we see is that that population that's over the age of 65 and comprises the vast majority of breast cancer patients is actually only accrued at rates of about 20 to 24%. And these are analyses that were done by my colleagues here at the Farber, Rachel Friedman and Ann Partridge, some of which is published and some of which is unpublished. So I thank them for sharing with me. If we look also at the younger age group, those women that are under 40, while they represent small numbers, there are significant numbers of women of young age who have breast cancer and unfortunately die from breast cancer. They too, because of those numbers, are accrued at very low rates, and they actively enroll in studies, but their numbers are lower, such that together they're underrepresented in clinical trials. And so here's what's particularly striking about those age groups that I just mentioned. And this is analysis that Rachel Friedman had also looked at across stage and subtype. If we look at the age extremes, and here's just the numbers staring at you, that those women tend to do worse. If we look at the adjusted hazard ratios for breast cancer specific survival by age, that the women at the ends of the spectrum tend to do worse than their middle-aged counterparts. And here's a more striking visual of that Dr. Friedman had put together, that regardless of stage or subtype, the women's at the end of the age spectrum do worse. Unfortunately, we know that stage 4 patients, for instance, tend to do worse overall. If we focus particularly now on triple negative breast cancer, which will be the focus of the work that we're doing in my lab, and we look at triple negative breast cancer, which accounts for about 15 to 20% of breast cancer cases, unfortunately lacks targeted therapies, we see that the distribution of triple negative breast cancer by age is fairly equal across the age spectrum. However, if we look at distant disease-free survival by age, younger women tend to do worse than their middle-aged or older counterparts. And that led us to really think of two straightforward questions. And really, we're wondering, does age affect breast cancer progression? And secondly, does age affect response to breast cancer therapy? And to do that and model that in our laboratory, I need to take you back to the history of our lab and how we sort of landed on this area of study. And that's back over a decade now, looking at breast cancer as a systemic disease in a process that we term systemic instigation, but it is well developed and understood now by our lab and a number of labs across the globe, which describes a powerful interplay between primary tumors and not only their local microenvironment, but the entire systemic microenvironment and organs at distant sites. And here I'll just represent that work in cartoon form and take you through some of what we know. As I mentioned, we're looking at various preclinical models of triple negative breast cancer in the lab, here represented by this cartoon image. And what we learned is that primary disease, primary triple negative breast cancer in our preclinical models secretes a myriad of factors into the local microenvironment and into the systemic environment, into the circulation. One of those factors is the growth factor osteopontin. What do we know about osteopontin? There's actually a huge literature on it. It's a pleiotropic growth factor secreted primarily by epithelial cells, but also by monocyte macrophages. It's known to regulate immune function, trigger inflammation. It has a number of properties, but what I really want to focus on is represented here, particularly these are the breast cancer data, but osteopontin is secreted at high levels into the circulation. It's detected in the plasma at high levels of patients of all types of cancers. If we look at the breast cancer data, particularly across the board, we see that high levels of osteopontin in the plasma are found in patients with metastatic disease relative to cancer-free women or patients without metastatic disease. And here, if we look at the cumulative portion surviving over time, unfortunately, those high levels of osteopontin in the plasma are correlated with reduced survival. Importantly, this will come up later, is that plasma osteopontin levels do not seem to be correlated with age or menopausal status, but primarily triggered by the presence of disease. What we learned at the time is that osteopontin was necessary but not sufficient to activate a subset of cells in the bone marrow that we identified by the cell surface markers SCA positive, c-KIT negative. And those cells in the marrow are activated to turn on CSF1R receptor and granulin. And I'll have to say right now, you may be wanting me to describe those cells, but we don't really have a handle on the identity of those cells yet. It's under active investigation. So, but what we found is that osteopontin, again, is necessary but not sufficient to turn on granulin, CSF1R, among a whole host of genes in this population of bone marrow cells. And what we know about granulin at the time was actually not quite much. It's expressed primarily by hematopoietic cells, particularly plasmacytoid dendritic cells, some epithelial cells, and some neurons. It mediates inflammation and wound healing, but at the time, its cognate receptor hadn't even been identified. It's thought to be a very primitive growth factor that's conserved all the way back to slime molds. But what has been understood since then is that it's highly upregulated in aggressive cancers, including breast cancer. It's mutated or lost in some neurodegenerative diseases. But what more recent work has shown from a number of labs, including Hitaplu's lab, looking at, granulin, a function for granulin, and bringing CpG DNA to the TLR9 receptor to modulate immune responses. And that may or may not become important to what we're studying in our laboratory. What we had observed is that these granulin high CSF1R positive cells do indeed mobilize into the circulation in response to osteopontin. And those cells can land at metastatic sites in distant tissues. And here I'm representing indolent disseminated tumor cells by this pink cluster in the metastatic microenvironment. And what we found is that granulin in those metastatic sites actually activates fibroblasts. Normal tissue fibroblasts are activated to turn on a whole host of genes, including very pro-inflammatory factors, alpha smooth muscle actin, so they look like myofibroblasts, and a whole host of genes that would be classified as pro-inflammatory genes, mediated again by granulin. Zafira Castaño, when she was a postdoc in the lab, also identified that these bone marrow-derived cells secrete EGF and IGF-1 growth factors that directly impinge upon disseminated cells to activate their proliferation. And collectively, the systemic cascade of events serve to induce metastatic progression, again, in our preclinical models of triple-negative breast cancer. We had also done this work primarily in immunocompromised mice. So it told us that these cells, which again, we have yet to really identify and purify, are innate immune cells. So we think in this process, this is an important innate immune response, particularly to osteopontin, that triggers disease progression. And when we look more closely, Moshe Elkabetz, when he was a postdoc in the lab, he's now an assistant professor in Ben Gurion, he looked at a tissue microarray of breast cancer cases that was comprised by our colleagues in Uppsala and Frederick Ponton's group. And they had 3 different antibodies to granulin. And so on this tissue microarray, they stained for granulin using their 3 antibodies. They sent us the staining, and together with our colleagues at the Broad, particularly Ann Carpenter, we quantified the amount of granulin staining in those tissues. And I'm just showing you here an example of that staining, how we quantified granulin by looking at the total tissue area, the area that was high for granulin, and quantifying that staining. And so when he did that across the entire tissue microarray, we sent the staining results back in a blinded fashion back to our collaborators, and they then looked at the clinical pathologic features of the tumor tissues from patients in that comprised that array, and found that granulin was highly associated with triple negative disease and inversely correlated with luminal. It turns out that actually their numbers of triple negative in the array were lower than the numbers of luminal breast cancers, but it was still very highly correlated with the triple negative breast cancer cases, and unfortunately correlated when examining cumulative survival those patients with tumors with high granulin positivity had worse survival in this case. So we could also do a number of tricks in the lab that we wouldn't necessarily do in the clinic, but to help us understand this systemic process, we could either surgically resect the primary triple negative breast cancers that instigated this entire cascade of systemic events, or we could transplant the very early lesions that had been exposed to the systemic environment initiated by the primary tumor and transplant those small early lesions into new host mice and find that kickstart, if you will, of systemic instigation was sufficient to enable these tumors to now grow very well on their own, when otherwise in control animals, they would not grow as rapidly. So it said to us that this process at some point becomes irrelevant for those disseminated tumor cells, but that early exposure to the primary tumor and the systemic cascade that it instigated is very important. And we use this now as a platform for identifying ways that we may perhaps interdict the systemic cascade to eventually prevent the outgrowth of or progression of disease. And that's serving as a platform in our lab now that we would hope we can make some inroads with, especially if we look at clinical correlates. But what I want to tell you about now is essentially a failed experiment. We were getting ready to, we were just gotten reviews back from a paper, and we needed to do a couple more experiments in our systemic instigation model. And we didn't have any mice on hand. It was a quick experiment that we just needed to do. Didn't have any mice on hand. Turned to a colleague who had mice in the same facility, and he said, "Oh, well, we've got some mice down there. You're welcome to use them. We're not using them now." So we set up our experiment, and it failed. We were looking for systemic instigation to characterize a little more deeply what was going on in the microenvironment, but we didn't get any disseminated tumor growth. So we were wringing our hands, worried about what was happening. We had done these experiments over and over, many times over many years, and had seen it very reliably reproduced, except in this one instance. And it turns out we looked at the cage cards of the animals that our colleague had given us, and they were 1 year old at the time they were enrolled into our study. So that set off some triggers that actually were in some ways very fortuitous, that led a group of people in my laboratory, Dr. Jacqueline Sini, a former postdoc in the lab, Timothy Marsh, who was an undergraduate at the time and now has his PhD from UCSF, Irene Wong, who was actually a high school student when she joined the lab and is now in graduate school right here at Harvard Medical School, about ready to get her PhD, and a wonderful technician, Amy Barakat. And they asked then, given our focus on this subset of immune cells that is responsible for instigating disease progression, they asked whether hematopoietic or immunological age affects triple negative breast cancer progression. And here's just a very, very simple but straightforward observation that is well understood. We know that if in humans, if we look over a period of time, that our immune function peaks when we're quite young, actually in our 20s, 30s, and starts to decline with age. That's something that's well observed and understood. But unfortunately, with age, we also see increased incidence of diseases, infectious diseases, autoimmune diseases, and cancer. What we know happens particularly in the hematopoietic system and the immune system, and I'm summarizing here, are a myriad of effects that essentially is with respect to hematopoiesis. We see myeloid skewing, a bias toward myeloid differentiation in the bone marrow, and a decrease in lymphoid production and maturation. We see thymic involution. We know that we see a decrease in naive T cell pools, increase in memory T cell pools, enhancement of inflammatory cytokines, and overall changes in the immune system that lead to this decline in immune function that we see with age. And so keeping that in mind, when we think of the cohorts that are particularly of interest to us with respect to breast cancer, women under 40 fall into this category where their immune function is still in breast cancer free patients is still relatively robust, whereas in those over the age of 65, we start to see this decline in immune function in general. So we performed a series of experiments in our animals whereby we used mice that are 8 to 10 weeks of age. And I have to say that nearly all breast cancer modeling, at least, if not all cancer modeling, is pretty much done in young mice. And these equate to humans that are particularly around under the age of 40 years. But in our case, we aged our mice out to 12 months, representing immunologically old. We're not using geriatric mice that are very, very old, but we're using immunologically old mice that are greater than 12 months of age, anywhere from 12 months to 18 months of age, roughly representing humans immunologically that are about 60 years of age. And again, because we were focused on that innate immune population of cells, the majority of these initial studies that I'm going to describe to you now were done using human cell lines or PDXs in immunocompromised nude mice. And so because it's hard to enroll old mice into our studies, we essentially have to wait a year, buy the mice, wait a year for them to age. The first thing we do with those mice is and I didn't bring it up here, is we actually have a birthday party for the mice when they reach 1 year of age. So that alone is quite a feat in the lab to wait that long to actually do your experiment. But what we had observed, and when we inject the exact same tumor cell line, in this case, these are BPLER human tumor cell line developed in Bob Weinberg's lab, we inject that exact same cell line into either young or old mice and see right away that in the young mice, those cells progress quite rapidly. Whereas in the older mice, there's a longer latency period before they start to grow, and they grow much, much more slowly. And young and old mice, and here I'm showing you the histopathology of those tumors that arose in those animals. We see that proliferation as measured by Ki67 in those tumors, as well as granulin, that important granulin derived, the granulin expressing bone marrow cells are decreased in the tumors of the older animals compared to the younger animals. So it already gave us some hints that something was very different, even though we had injected the exact same tumor cells into these mice. The age of the animal made a significant difference in the way that those tumors progress. So we did another set of experiments where we allowed the tumors from both young and old mice to attain the same size. So now we're doing a size-based experiment where we look at both young and older animals in which the primary tumor is the same size that we do our analyses. We look at plasma osteopontin in the blood and find that there's really no significant difference. Indeed, we see an enhancement in the presence of tumors, but that enhancement is equal in both the young and aged animals. But what was important is that the incidence of distant progressive disease was significantly lower in the aged mice relative to the young mice. So even though primary tumors had attained the same size, plasma osteopontin is the same, we see that the incidence of disease progression is lower. And that's something important because it harkened back to the clinical data telling us that disease progression is more rapid in younger women. We look again at the tumors that had formed in this case, and we see again the young tumors, infiltration of granulin positive cells, these SCA1 positive cells, is high relative to the older animals, and here the data are quantified on the right. And when we look at the bone marrow, looking for the cell type of interest to us, we see that actually with age, that SCA1 positive c-KIT negative cell population actually expands with age, but there's no tumor dependent effect on the numbers of those cells in the marrow. So again, whether the mice are young or old, the tumor doesn't make a difference, but age makes a big difference, and actually an expansion of that cell type in the marrow. Again, we think it's an innate immune cell type, and this would correlate well with myeloid skewing in the bone marrow. If we look more closely at that population of cells in the marrow that's important to us, and we look for granulin positivity and CSF1R, we do see that indeed in the young mice, tumors enhance the levels of both granulin and CSF1R, and these are now relative to the cancer-free young animals. However, we don't see the same in the older animals. In the older animals, there's no increase, that tumor-dependent increase in granulin or CSF1R on those cell types, those innate immune cells that are of interest to us in the marrow. But here's the real, what I call the clincher experiment, that really convinced us that hematopoietic and immune age really do matter in this context. So I'll walk you through these experiments. What we had done is we had taken young mice bearing triple negative breast cancer, again, our 8 to 10 week old mice, instigated tumors in those animals, triple negative breast cancer model. We isolate their bone marrow, and we immediately mix that marrow with indolent tumor cells that grow very slowly. In this case, these were HMLER cells derived in Bob Weinberg's lab. We've done it using a number of different cell lines that actually grow slowly. Our goal was actually to look at increased growth. So we use very indolent tumor cells. Again, mixed it, just an admixture with bone marrow cells that were derived from tumor-bearing animals, and immediately inject those mixtures into either young or old cohorts of mice. It's a readout for how functional that bone marrow is in supporting tumor growth. So we can do that, and we can also take the marrow from older mice that have triple negative breast cancer. In this case, again, we size match those primary tumors, isolate their bone marrow, mix the marrow with the same set of tumor cells, and inject those mixtures either into young or old animals. And what we observed, and we had observed this in our previous studies and repeated here again, is that we can take the bone marrow from a young tumor-bearing animal, mix it with indolent cells, and inject those mixtures into young animals, and we see, with time, we see rapid induction of disease progression. Here you're looking at the percent tumor-free as a surrogate for survival in our animal models. But if we take the marrow from older animals with triple negative breast cancer and we do the same admixture, no matter whether we inject those into young or old animals, we don't see the same enhancement of tumor growth that we had observed when we used the young bone marrow. Importantly and quite strikingly, when we took young donor marrow, did the admixture, and put those tumors now, that admixture, into old animals, we see a very enhanced tumor growth in those older animals. So this said to us that hematopoietic age is really important in terms of its tumor support function, and that with age, that tumor support function declines. And so we've isolated those, again, SCA1 positive, granulin positive, CSF1R positive cells from the marrow, and did a deeper dive doing gene expression array analysis at various points here, either in cancer-free animals that were young or old, or in tumor-bearing animals that were young or old, and looked at gene expression profiles as a platform to understand what's going on in tumor and age-dependent ways. And we see whole hosts of, as expected, whole hosts of pathways that are regulated in both age and tumor-dependent manners. I'm not really going to highlight anything here because we're using these data to move forward to try to understand general processes in this subtype of cells that's tumor supportive. that are altered again in tumor or age dependent ways. And so that's future work in our laboratory. So we're starting to think now of pro and anti-tumor effects, at least with respect to the results that we've observed. And if we consider, again, just the pro-tumorigenic function of these granulin positive cells in the marrow, we see that the weight is shifted in young animals quite significantly toward pro-tumor immunity, whereas in older animals, that weight is a little smaller. And we think of that now, then it seems that there's much more to overcome in younger animals than there is in older animals to perhaps shift that balance. And that's important in our thinking. And again, we want to then model the situation where disease does seem more aggressive in younger women. Is this one little aspect that we've uncovered an important player in that, and that there's more to overcome in younger animals or younger women. But what about the anti-tumor immunity side of the equation? How much weight is actually on the scales in that context? And so that's a question that was asked by, again, Dr. Jacqueline Sini when she was a postdoc in the lab, and along with Greg Goresny, who also is a former postdoc in the lab. They were asking that question of, does age impact anti-tumor immunity? And in this case, particularly looking at response to immune checkpoint blockade in triple negative breast cancer. And again, we use our young and age models. In this case, we're going to focus now on one particular model, and that is the 4T1 cell line that was derived in Fred Miller's group. It's a triple negative murine cancer cell line derived from a spontaneously arising memory carcinoma. And that cell line can be injected into syngeneic BALB/c mice. And I just want to thank the NIA, because we did acquire 1-year-old BALB/c mice from the NIA, which saved us the trauma of having to age mice in our facility. We still have a birthday party for those mice too, though. Just to check that we are dealing with an appropriate age model, Jacqueline and Greg looked at circulating immune cells and looked at the peripheral immunity in the aged and young BALB/c animals. And we do see, as expected, that myeloid skewing in the marrow and in the blood that occurs with age, and a retraction of the lymphocyte populations. When they look more closely at both blood and spleen, first in the blood, they do see a decrease in the total numbers of CD8 and CD4 positive cells. And in the spleen, there's a particular expansion in the older animals in both CD8 and CD4 positive populations of central memory and effector memory cells, and somewhat of a retraction of naive memory cells, particularly observed in the CD4 positive population from the spleen. But now here we do our tumor initiation studies, and we inject those 4T1 positive triple negative breast cancer cells into the memory fat pad of the animals that are young or old. And here we have a model whereby there's not a significant difference in the tumor growth kinetics in the young and old animals in this case. So that primary disease is actually not affected, or doesn't appear to be affected by age, at least as it manifests in tumor growth. They were particularly asking the question of evaluating immune checkpoint blockade therapy, monotherapy, in the young and old animals. As you know, at this period of time, immune checkpoint blockade had a number of successes in the clinic, particularly for melanoma and non-small cell lung carcinoma. However, other diseases like breast cancer that's thought to be not so immunogenic took a while for us to start bringing that into the clinic. But we're now, seeing quite a number of trials that are evaluating immune checkpoint blockade, particularly for triple negative breast cancer and other cancers that were thought to be less immunogenic. So they initiated a study in the animals to look at how immune checkpoint blockade affects triple negative breast cancer in young and old animals. And here's the study schema where they inject the tumor cells, look at 4 sequential doses of either anti-PD-L1 or anti-CTLA-4 or an isotype control in young and aged animals. And what they observed is that in the young isotype treated control, tumors grew as expected in the young animals. Anti-PD-L1 monotherapy did have a significant impact on tumor growth, and the mice benefited in terms of a survival benefit in those animals. But now looking at the young animals, isotype and anti-PD-L1 really grew, tumors grew really to the same extent in those animals, and there was no survival benefit imparted by that monotherapy for the older animals. Likewise, they did the same with CTLA-4, which seemed to be much more effective in the young animals and imparted a significant survival benefit in the young animals. But again, with age, we didn't see any difference in tumor growth or final tumor weight. And again, no survival benefit imparted by anti-CTLA-4 treatment of the old animals. And our colleague here at the Farber and the Brigham, Dr. Beth Mittendorf, had established that evaluation of PD-L1 and tumor infiltrating lymphocytes is clinically meaningful in breast cancer, and particularly triple negative breast cancer. So we undertook similar studies in our animals to understand those effects. And first of all, we did confirm in this 4T1 tumor cell line that they indeed respond in vitro to interferon gamma by upregulating PD-L1 and MHC1 in this case, telling us that those tumor cells are capable of responding to interferon gamma stimulated effects. We also then took a closer look at the histopathology of the tumors that formed to assess tumor infiltrating lymphocytes, in this case CD8 positive cells, and to categorize them into the well-known categories now, either as immune deserts where no CD8 positive cells are observed, or an inflamed signature whereby significant numbers of intratumoral CD8 positive T cells, here indicated by the red staining, infiltrate the tumors, or classify them as borderline, whereby T cells are observed in the tumors, but they're restricted either to the stroma or the periphery of those tumors. And here, when we classified them, it wasn't quite as straightforward as just these 3 subclasses, but we had tumors where we scanned the entire tumor tissue, in this case, not just a region of interest, and classified them as either cold, which would be immune deserts, or hot, where we see significant infiltration of TILs in our preclinical model. And what we see is that in the young animals, that those tumors treated with either anti-PD-L1 or anti-CTLA-4 do become more infiltrated with tumor infiltrating lymphocytes. However, when we look at the old animals in a similar fashion, we see that there's not an infiltration of CD8 positive TILs. And if anything, they almost seem to get colder. And so we came up with the slogan that, in this case, old stay cold. We're not seeing a significant infiltration of CD8 T cells. And here, Jacqueline particularly did a significant number of analyses on peripheral blood, tumor, and spleen tissue by flow cytometry to understand the composition of the immune repertoire. And here I'm just going to try to summarize the main points from going through all of these data. And what they had observed, particularly in an age-dependent fashion, there's an increase in circulating memory cells, CD4 and CD8, and PD-L1 positive CD8s, but a reduction in naive CD4s with age. Again, in the age-dependent fashion, there's an increase in the naive CD8 cells only in the young animals treated with anti-CTLA-4, not observed in the older animals. and pardon me. And likewise, we did see an increase in PD-L1 positive CD8 and CD4s only in the context of anti-CTLA-4 monotherapy in the young animals. When looking at the spleen, again, in a striking way, seen in an age-dependent fashion, it was not necessarily affected by anti-PD-L1 or anti-CTLA-4 in the CD4s, was an increase in checkpoint proteins, particularly PD-1 and TIM3. Whereas in the CD8 positive population, with age, again, there's an increase in those checkpoint proteins on the CD8 cells that's somewhat enhanced with treatment, particularly PD-L1 in the splenocytes. And here's where we started to get some clues about what was happening when we particularly looked at changes in cytokines and transcription factors on the CD8s and CD4s in the peripheral immune microenvironment. And this is looking at the tumor immune microenvironment. So in the tumors themselves, looking at CD8s and CD4s, seeing an age-dependent decrease in interferon gamma in the CD8 positive cells, quite striking reduction, just looking at the isotype treated animals where there's already a reduction in interferon gamma that's not enhanced by therapy in the aged animals as it is in the young animals. And taking a closer look now at absolute numbers of CD8s by flow cytometry, looking at interferon, again, a significant decrease just in the baseline levels with age that are not increased with therapy in the older animals. And I threw this in for those who are interested in looking at monocyte macrophages. The total number, I'm sorry, I keep hitting that wrong button. The total numbers of F480 in the tumors is actually decreased in old. So that's something that requires further investigation. And now to take a deeper dive into this, we set up gene expression analyses to look at the tumors of these animals in these various cohorts, young and age, isotype treated or treated with monotherapy. And what we see right away that was very striking to me, again, this is gene expression profiling of the tumors from young and old animals. Just looking at this pattern, it says to me that, at least in these preclinical models, triple negative disease is very different in young and aged animals. If we take a closer look at gene set enrichment, the genes pathways that fell right to the top of the list with treatment here, comparing now young isotype to PD-L1 treated or young CTLA-4 to the isotype treated animals, is an enrichment of inflammatory responses, interferon gamma, suggesting that we are having a sufficient immune response triggered by these checkpoint blockade proteins. But that is not true in the older animals. These same pathways were not enriched in the animals treated with immune checkpoint blockade. So that was our first suggestion that typical, triggering or immune priming may be impaired in these aged animals. And this is importantly also observed if we just compare the young isotype to the old isotype, untreated animals, essentially, we see that really at baseline, there's already an enrichment of type 1 interferons, as well as interferon gamma and inflammatory response pathways that already at baseline are much higher in younger animals than older animals. So that gave us an opportunity, again, because immune checkpoint blockade hadn't been evaluated very much in the breast cancer clinic at this time. It gave us an opportunity to turn to our collaborator, Dan Stover, who's now at Ohio State University, to analyze a Metabrick data set for triple negative breast cancer stratified by the age groups of interest, those under 40 and those over the age of 65. And again, if you just look at this pattern, it says to us that triple negative breast cancer looks like a different disease in younger and older women. And what he found, he had not known our results from our preclinical models. He did this blindly. And what came to the top of the list when he did gene set enrichment analyses of those patients were interferon gamma responses, interferon alpha type 1 interferons, enriched hallmark inflammatory response pathways, JAK-STAT signaling, IL-6 signaling, pretty much the same types of immune pathways that we observed in our preclinical model are being enriched in younger patients under the age of 40 versus those over the age of 65. So indeed, it said to us that triple negative disease from an immune perspective looks very different in younger and older women, at least in this MetaBrick data set. So that got us, fortunately, there are ways to stimulate interferon signaling, and that's through the use of STING agonists that are also now being evaluated in the clinic to try to stimulate immune priming here. STING is very important for the sensing of tumor-derived DNA. It will trigger the induction of type 1 interferons to then eventually enhance cross-priming of tumor-specific CD8 cells. And when we can use a STING agonist and evaluate that in our preclinical models of young and older animals in a dosing regimen that I've outlined here, using the STING agonist DMXAA in combination with anti-PD-L1 or anti-CTLA-4 therapy. And what we see in this focus just now on the older animals, in those older animals that had no benefit with either PD-L1 or anti-CTLA-4 therapy, the combination of DMXAA, the STING agonist, did improve survival in those animals, in the older animals, either with combination of PD-L1 or anti-CTLA-4. So again, a significant survival benefit imparted by that combination therapy in the older animals. Importantly, we didn't see, I'm not showing you here, but we didn't see an additional enhancement of benefit in the young animals. It seemed like they were already maxed out with anti-PD-L1 or anti-CTLA-4 such that DMXAA didn't add any benefit. So it's very specific to the older animals. And so when we think of now our balance in this context, we think of the fact that already in young animals and also with the clinical correlates from the MetaBrick data set, already at baseline, there's more weight on the scale of anti-tumor immunity in young, such that here I'm just lumping them together as interferon responses are already sort of tipping the scales toward anti-tumor immunity. The addition of immune checkpoint blockade adds weight, even bigger weight, in the young animals, again, to tip the scale even more toward anti-tumor immunity, whereas in the old, that weight is slightly less. We're not seeing the same robust responses. However, we can add the STING agonist to the scale in the older animals and see now that we've indeed tipped that balance that does impart a real biological survival benefit. But STING in the young animals didn't carry as much weight, and we couldn't tip the balance anymore in favor of anti-tumor immunity. So what about the breast cancer trials? What can we learn, or are we learning anything about age in response to checkpoint blockade therapy? Again, only relatively recently are we seeing checkpoint blockade therapy evaluated in clinical trials. And here I'm just showing you a summary of some of the initial monotherapy trials looking at here anti-PD-1 pembrolizumab or avelumab, PD-L1 or atezolizumab in monotherapy in either metastatic triple negative or here hormone receptor positive disease. And what if we try to look at age in those studies? Well, as I explained in the very beginning, unfortunately, women at the age extremes that we're interested in, under 40 and over 65, the numbers just aren't sufficient to be able to evaluate whether age is where there are differences in response with respect to age. However, this one trial, the Keynote 086, there were sufficient numbers of women. Here they stratified by under the age of 50 or over the age of 50 to observe a trend. And at least in this case, what we're seeing is a trend toward benefit of the pembrolizumab in the young relative to the older women. And here again, just stratified by over and under 50. Now, what about chemotherapy backbones? There's now a lot of efforts going on to understand what chemotherapy backbones may be sort of providing a platform for better response to checkpoint blockade therapy. And one very successful trial that's was conducted by our collaborator and friend here, Dr. Beth Mittendorf, just reported some very promising results looking at evaluating atezolizumab in the neoadjuvant setting for early triple-negative breast cancer in the IMpassion031 trial. And here in this study, they're evaluating atezolizumab with a backbone of either nab-paclitaxel or chemotherapy, doxorubicin and cyclophosphamide, and looking at pathological complete response at the time of surgery as one of their co-primary endpoints. And recently just reported that regardless of PD-L1 status, there is a benefit of atezolizumab, again, regardless of PD-L1 status, in pathological complete response. So Miloš Spasek, who is a PhD, who is a postdoctoral fellow in my lab currently, essentially set up a preclinical study to mirror Dr. Mittendorf's trial, using now a MET1 triple-negative breast cancer cell line that was derived by Sandy Borofsky. It's a cell line developed from the polyoma middle T on an FEB mouse background. So we can inject these triple-negative breast cancer cells into these FEB mice. And we do indeed confirm that those cells in vitro are capable of responding to interferon gamma by upregulating PD-L1 and MHC1, and set up a trial to look at now with a backbone of paclitaxel, adding anti-PD-L1 treatment to the therapy. And I'll just take you through some of the important points. This is all unpublished data that's fairly new from Dr. Spacek. And what he observes, first of all, just in the isotretted animals, is that tumor growth, as we've seen in other models, is actually slower in the aged animals. So here's what we think is a good model to understand that progression of disease with age, that in younger animals is quite rapid. But what he observed that's important here, if we look at the clinical correlate, is that paclitaxel combination therapy with anti-PD-L1 imparted a significant benefit. You're looking here now at the magnitude of response, if you will, that anti-PD-L1 therapy imparted a significant benefit to paclitaxel backbone in the young animals, but not in the old animals. So here he's conducting these studies now and wants to understand what may be, again, responsible for the lack of benefit that was observed in the older animals relative to the young. And so what if we look at Dr. Mittendorf's trial for age? And here they did report age groups under the age of 40 and here over the age of 40. And looking at a trend, again, the numbers aren't quite significant, but looking at a trend, it seems that the younger women, if we look here, if we shift to the right, that means that the atezolizumab imparted benefit over the placebo. And here, if we look at the trend, younger women are tending to do better with the addition of atezolizumab than the older women. So, we're talking with the trial investigators to be able to take a closer look at age and maybe stratify into the age extremes and look at how can we look at age in these clinical trials. One of those clinical trials is the IMpassion130 that was reported out to show a significant benefit in a PD-L1 positive population in the metastatic triple-negative breast cancer setting when atezolizumab is added to a backbone of nab-paclitaxel. And this study did report age stratification in a way that was helpful to look at the age extremes. And here in this case, in the metastatic setting, it looks like the older women may be doing somewhat better with the addition of immune checkpoint blockade therapy, atezolizumab in this case. So again, we'd like to talk to the trial investigators to take a closer look at these age considerations. So now when we bring the picture back together, we think of this balance between pro and anti-tumor immunity, and it's clear, at least from our studies, that pro-tumor immunity and anti-tumor immunity in young both carry more weight. There may be much more to overcome in young with respect to shifting that balance from pro-tumor to anti-tumor, but there's already some weight on the scale of anti-tumor immunity, which we don't observe in this case, the older animals. So what kinds of efforts can be made to really understand age? And again, with that problem or that challenge that older and younger women just aren't represented in clinical trials as much. And I really want to stress the efforts here, right here at Dana Farber, of two of our collaborators and colleagues and friends, Dr. Ann Partridge and Dr. Rachel Friedman, who are looking at caring for both the younger and the Young Strong program, of which Ann Partridge directs, and this newer program of studying older patients and trying to enroll them in trials and studies to turn silver into gold, led by Dr. Friedman. And together with Dr. Friedman, we formed the Older Women with Breast Cancer Research and Treatment team right here at the Brigham and the Farber to begin to analyze data, clinical data, as well as research, and bring a sort of a bedside to bench to bedside approach toward understanding age and how it affects disease progression and response to therapy. And so with that, I really want to thank the tremendous team of people with whom we work, past and present members of my own lab that I have listed here in bold. Those are the particular the people who worked on this aging study. I really want to shout out to the breast cancer advocates at the Dana-Farber Harvard Cancer Center, led by Liz Frank, and particularly Barbara, Christine, and Margaret, who were actively involved in, from writing the grant to help writing papers, to really interact with us and help us think about our work in clinically meaningful ways. And our amazing team of breast cancer surgeons and oncologists here at the Farber, with whom we work closely, and a number of people around the world who have all contributed to this work. So with that, I will thank you, and I don't know if there's time for questions. I hope there is. And if not, I'll try to answer some things online.
Thanks, Sandy. This was an amazing talk. Thank you so much. And we have a number of questions. We kind of running out of time, but let me just at least pick some that relate to them we could answer, and then you could you get the list of others as well. So Jim DeGregory and Patrick Anderson, both of them were asking related question that, wound healing and angiogenesis in general also seem to be, diminished in an older age. Could this have something to do with the young-old difference? Have you looked at the vascularization of these tumors? Maybe the reason the cells cannot get in because you don't have as many blood vessels. So that's one of the questions.
Yeah, I think, hi, by the way, hi to everyone. I wish I could see you. James, I know we'll be seeing you soon. It's a great question, and we don't have an answer to it in our models. We can answer that question, and we're glad to work with anyone who wants to really take a deeper dive into that. Again, we've seen those changes in fibroblasts with age. A number of people, Ashi Weeraratna, who gave a great talk in this series earlier, upcoming Sheila Stewart, looking at fibroblasts in various tissues with age. I think that will become something very important. Again, we saw those changes. We didn't really look yet at vascularization. I think these are all things that are ripe for us and collaborators to understand. Yeah.
Another question, whether you looked at the Tregs. Do you see a difference in Tregs in the old and young animals and people if they looked at?
Yeah, that's interesting. In the first model I talked to you about, where we looked at the 4T1 tumor cells injected into young and old animals, we didn't necessarily see striking changes in Tregs. It seems we may be it seems it may be model specific, where some Tregs are infiltrating more than in other models. And that's something we, again, want to take a very close look at.
Okay. Another question is that, this age difference in women, do you think that also could be influenced by the hormonal levels? So have you looked at, within a cohort, not just the age, but what is their hormonal status, maybe BMI, and, looking at immunological age, which is not always identical with chronologic age, like people who exercise maybe like 10 years younger than their age. So that could be, the interdisciplinary human studies could influence the differences.
Absolutely. Hormonal age is something we really need to consider, obviously, in our models. We haven't built them necessarily into our mouse models. We're focusing on triple negative. Not that hormones aren't important in that context, but yes, immunological age is something we really, really are taking a very close look at. There are some beautiful studies, and actually I could put the paper up on the link or share the paper with anyone. A beautiful study done, and I'm sorry, I'm blank right now on the authors, but looking at immunological age, not necessarily in the context of cancer, but essentially following people for a very long period of time and looking at intra and inter-individual differences, and defining an immune age signature that doesn't necessarily correlate with chronological age, but does correlate with onset of various types of diseases. So that immunological age, I think, is absolutely critical, and something we want to try to go for in our modeling and clinical correlates to determine whether we can define that a little in a slightly better way, so that we don't have to necessarily look at over 65 and under 40. We can look across the board, define that, and make some predictions on outcome or perhaps response to therapy. So yeah, great question.
Okay, thank you so much. I think we ran out of time, so we'll wrap it up now. Thanks again, Sandy, and thanks everyone for listening. And then again, the talk will be uploaded to the website, and the questions we will forward you to get back to the people, because we didn't have time to answer all of them. So thank you so much.
Great. Thank you, everyone.